Hello, my name is Paige Gott. I'm a ruminant technical manager for Biomin, and I'm going to give a brief introduction to mycotoxin risk management. What are mycotoxins? Mycotoxins are secondary metabolites produced by fungi. Over 400 different types of fungi have been identified which produce mycotoxins, with more than 1,000 different mycotoxins being identified. 25 to 30% of the world's crop is contaminated with mycotoxins according to the Food and Agriculture Organization, or FAO. 95% of mycotoxins are produced while the crops are still in the field. So not often when we talk about mycotoxins and the molds that produce them, we've classified them as field fungi or storage fungi. And their names are pretty descriptive, but the field fungi would be those that are growing in the field and producing toxins prior to harvest, whereas the storage fungi are molds that would be growing and producing toxins after harvest. There are exceptions, during hot or dry conditions where the different molds are able to survive during the different time periods and produce toxins beyond when we would normally expect it. So some of the field fungi can survive and continue to produce toxins after harvest when the crops are in storage, and some of the fungi are able to grow in the field and produce toxins prior to harvest. The two different groups, so when we look at the field fungi, we really focus on the Fusarium species, which is capable of producing multiple types of toxins, as you can see here in this chart. So on the right-hand side, we have little graphics or pictures of the different types of fungi, and then on the left-hand side would be the chemical structure of the mycotoxin. So Fusarium is the type of mold, and fumonisin would be the group of toxins. And looking at the storage fungi, we really talk about the Aspergillus species and Penicillium species as being storage fungi. When we talk about mycotoxins, there are 6 main types that we often focus on. Those would include aflatoxins, zearalenone, ergot alkaloids, fumonisins, trichothecins, and ochratoxin. There are multiple different toxins that fall into each of these categories. For instance, the trichothecins, there are more than 200 trichothecins that have been identified. So the toxins are grouped based on their chemical structure and often the molds which produce them. So you can see in this picture 2 of the aflatoxins that I have structures shown here. You can see how similar their chemical structures are. Looking at trichothecins, within the trichothecins, we categorize those into multiple different groups, including type A and type B trichothecins. What's similar among the whole group of trichothecins is this structure here. It looks kind of like a triangle with an oxygen as part of it, and that would be an epoxide ring. So all of the trichothecins have this epoxide ring as part of their chemical structure. Mycotoxins are important in livestock production because they can have negative effects on animal performance and production. And they do this through many different ways. Different toxins are associated with different negative effects. There is overlap, of course, but some of the different signs that we'll see are digestive disorders, including altered rumen function in cattle, diarrhea is seen in multiple species. Toxins can lead to immune suppression in animals, so they're less capable of fighting other secondary infections such as bacterial or viral conditions. Neurotoxicity, hepatotoxicity, some of the toxins are associated with kidney damage as well. The aflatoxins are considered to be carcinogenic, which is a concern, and that's why the aflatoxins are the one group of toxins which are regulated by the FDA. Zearalenone is associated with impaired reproduction primarily in females, but it can also negatively impact males. And overall, like I mentioned, these toxins lead to decreased performance and production. This graphic focuses on cattle, since that's what I primarily work with, but we do see similar effects in all of the different livestock species, and even companion animals can be negatively impacted by mycotoxins. And the top line of each of the blue boxes, you'll see the different toxins that are associated, and then the bullet points are different signs that you would potentially see in animals. So like I mentioned, zearalenone is a reproductive toxin. It has a structure similar to estrogen, and because of that, it can interact with estrogen receptors, leading to these different effects that we would see, including irregular heats in females, low conception rates. There's also embryonic loss and abortions associated with zearalenone. Like I mentioned, it can negatively impact males as well, leading to underdeveloped testicles and low sperm production. There's a variety of different signs that we would see, anywhere from laminitis or lameness to decreased feed intake and feed efficiency. The ergot alkaloids are associated with impaired thermoregulation, so that can cause cattle that are exposed to heat stress situations that can exacerbate that heat stress and the signs that we would see because the cows aren't able to regulate their body temperature because of the toxins as well. A variety of the toxins lead to the different gastrointestinal signs, such as gastroenteritis, intestinal hemorrhages, altered rumen function, and diarrhea, like I already mentioned. One concern with dairy cattle is the potential of passing toxins through the milk. Like I mentioned, aflatoxins are regulated in feed and also milk of cattle because they can be, aflatoxin B1 is transformed into aflatoxin M1, which is passed through cow's milk. In addition to the possible toxin contamination, cattle might produce less milk and have more cases of mastitis. Mycotoxins are a concern around the world, not just in the US. One thing that Biomin does annually is a mycotoxin survey, where we collect different feed samples from around the world and analyze them for the presence of mycotoxins. This slide just shows some of the results of the 2015 survey of the US corn crop. More than 300 samples were collected from throughout the US. And you can see the DON, or deoxynivalenol, which is also commonly called vomitoxin, was found in 79% of the corn samples. The fumonisin were detected in 59% of samples, and zearalenone was isolated from 14% of the samples. This pie chart shows the prevalence of multiple mycotoxins. So 6% of the samples, no mycotoxins were detected. From 44% of the samples, one mycotoxin was detected, and from half of the samples, more than one mycotoxin was detected. So we mentioned the different molds. The Fusarium mold actually is responsible for producing all three of these types of toxins. So often when you have that mold, you might get more than just one, because it is capable of producing multiple types of toxins, in addition to the potential of having multiple types of molds that would contribute to multiple mycotoxins that way. When we look at the threshold levels and when we would be concerned to see the negative effects in animals, we see that 71% of the zearalenone samples were above 100 parts per billion. And that's the level where we would be worried about seeing these negative effects come through in the animals. 19% of the DON samples were above the threshold, and 12% of the fumonisin samples were above the threshold. When we talk about the presence of multiple mycotoxins, so 50% of these corn samples had more than one mycotoxin, we can see synergistic or additive effects of mycotoxins when there's more than one. So in that case, the effects would be even worse than what we would suspect by just having the one toxin. So there's worse things that would be seen. So 1 + 1 does not equal 2 in this instance. We could have 1 + 1 = 4, or something like that is what we would be concerned about when you have more than one toxin, that you will actually have worse signs in the animals than what would be anticipated. So what can we do to try to counteract these toxins? One common method is adsorption of the toxin, so binding the toxins with some sort of substance, often a clay or mineral component that's able to bind up those toxins and help them pass through the animal's digestive tract without having their negative effects. A second method is biotransformation, which would include enzymatic deactivation of the toxins by altering their chemical structure, making them either non-toxic or a much less toxic form. And then a third component that is used is bioprotection. So that would be providing nutrients and different ingredients that can help boost the animal's immune response and help them better fight off the negative effects of these toxins. When we talk about absorption, this diagram shows the absorption efficacy. So from the lowest efficacy up to the greatest efficacy at the top of the triangle. So this is just to show that different toxins bind at different rates. So aflatoxins are a class of toxins that actually are bound really well by some of the different binding products, such as the clay and the mineral products I mentioned. Ergot alkaloids are the second best group at being able to be bound. And then we go down to DON and T2 toxin, which are the least likely to be effectively bound by binding products. This is a chart from a study that was looking at the efficacy of adsorption. So in this chart, we're looking at the percent of aflatoxins that were bound by different products. So across the x-axis, you'll see many different products that were tested. And in the graph, you'll see the blue bar represents the binding of aflatoxins at a pH 3.0. And then the red squares are representative of how well that product bound aflatoxins at a pH 6.5. So pH 6.5 in cattle would be somewhat similar to what you would see in the rumen, and then pH 3 would be a lower pH that you would see further along in the digestive tract, like in the abomasum. So that's important to keep in mind, because you can see within the different products, some bind really well at one pH and not the other, or some of them are fairly consistent, like this first product, over 90% of the aflatoxins were bound at both pHs. At this product here, you can see over 90% were bound at pH 6.5, which is really good, but then when you move to an area potentially in the digestive tract with a lower pH, only about 50% of the toxins were bound. So like I mentioned, in a cow, this could be toxins are really bound well in the rumen, and then the feed moves down the digestive tract, we'll see some of those toxins becoming unbound with the change of pH when it gets to a lower pH. I also want to point out, so in this red box, these would be inorganic binders, so the clay and mineral products like I was talking about. In the green box, we would have organoclays, so these would be some sort of organic binding product, maybe like a yeast product combined with an inorganic portion. And then the blue box at the end is the organic binder, so they could be yeasts or enzymes. And then the yellow box would be showing that although these products were considered organic binders, they actually had a very high ash content, which would be indicative of the inorganic compounds. So they bound quite well, as you can see with these ones, but it appears as though they're actually more of an organoclay, a combination product here. You can see the organic binders have much less binding efficacy in comparison with the majority of the inorganic products. This is a similar chart, just looking at a different toxin. So this would be the absorption of DON, which I showed you at the beginning with that triangle, inverse triangle chart, that DON was at the bottom of the spectrum of binding efficacy. And you can see that represented here with these same products across the bottom, and the best products are only about 25% binding of DON across the board, and it's actually much less for the majority of the products and both at the different pH levels. Again, looking at the absorption, this is from a different study, looking at the absorption of zearalenone, one of the other toxins that I mentioned. You can see across the board here, we also have very, very low absorption, with the highest at about 60% in one of the products. And again, this is looking, the green bar again has to do with the different pH. So pH 3, which would be similar to the stomach, and then here they're talking about the pH 6.5, which would be similar to saliva or intestines in animals. And you can see the difference between the two pHs also with zearalenone. Another method that I mentioned to deal with toxins would be biotransformation, which is often done by enzymatic detoxification, which would be altering the chemical structure of the different toxins. Like I mentioned at the beginning, the trichothecenes all have this epoxide ring structure similar throughout the group. You can use an epoxidase enzyme to go in there and cleave that ring and break it and convert the trichothecenes to a less toxic or non-toxic version of the compound by cleaving that epoxide ring. Looking at zearalenone, it also has a ring structure to it, and the esterase enzyme is able to cleave that ring, rendering zearalenone to be a less toxic form. And the nice thing with the biotransformation and the enzymatic alterations is that these are irreversible. Once you change that structure, it's not going to be able to move to a different pH, for instance, in the digestive tract and convert back into this active form. So that is one benefit of biotransformation over the adsorption that I showed you. A third component in some products includes bioprotection, which again, that has to do with boosting that animal's response to the presence of toxins. This can be done with phytogenic ingredients or ingredients that would be of plant origin. They can help with liver protection by stabilizing the liver cells and cell membranes. There are also phycophytic ingredients, which would be ingredients of algae origin. Algae, different types of algae contain a wide range of minerals and elements, in addition to vitamins and other ingredients like important carbohydrates and amino acids that can be all used to contribute to increasing the animal's immune response to the toxins. Biomin is a company that's based out of Austria in Europe, and their product Mycofix, which is not available in the US or Canada, the components of Biofix have been approved by the EU as mycotoxin deactivators. So that's the first and only company with this authorization for 3 of their different ingredients used in their Mycofix product. So the one is their specific bentonite clay component in the Mycofix has been shown to bind mycotoxins, like aflatoxins, really well. The second component, they have a biotransformation component, which is a bacterial source that produces the enzymes which can enzymatically alter some of the different mycotoxins, rendering them inactive. And they also have a specific enzyme. So this would be, it's really a recombinant enzyme that they're able to produce called fumzyme, which is able to transform the fumonisin toxins and render those inactive. So again, this is a product that's called Mycofix. It's not available in the US or Canada. So a comprehensive solution would be to combine those 3 different modes of action to work against the toxins. Again, that would be the adsorption, biotransformation, and bioprotection. So binding the toxins that are capable of being bound and helping those pass through the digestive tract without having the negative effects on the animals. Biotransformation, so physically altering the chemical structure of the toxins, rendering them inactive in the animal, and then also providing ingredients that can help support that animal that's under challenge and help boost their immune system. So that would be the best way would be to combine those 3 ways to work against toxins and help support the animal. My email address is page.gott@biomin.net. If you have any questions about the presentation or anything else related to this topic, feel free to contact me, and I appreciate you for participating and listening to this recording. Thank you.